Hi everyone! In this video we are going to be talking about Hardy-Weinberg equilibrium. One way that I think about this principle, I guess you could call it, is that it's sort of similar to Newton's first law of motion. So remember that law, or I don't know if you've even taken physics yet, but that law, I think everyone probably learned it at some point or another, says that objects in motion will tend to stay in motion and objects at rest will tend to stay at rest unless they're acted upon by an outside force. And in a sort of similar way, what this Hardy-Weinberg equilibrium principle says is that a population will have a consistent genetic makeup from one generation to the next unless there's a disturbing force that's present. So without some disturbing force, no evolution is going to happen, and therefore genetic variation, so genotype frequency and allele frequency, is going to stay the same over time. And I'm going to skip ahead to this next slide to talk about what sorts of things we mean when we say disturbing forces. So this slide is showing the conditions that need to be in place in order for Hardy-Weinberg equilibrium to be true, in order for Hardy-Weinberg equilibrium to apply. And these conditions are that the breeding population is very, very large. And depending on the source that you look at, it could either be infinitely large, some sources say, or just very, very large. But the upshot is that we don't want things like genetic drift, which have a greater effect on smaller populations, to have an effect. We want genetic drift to not be a factor. Another condition is that mating needs to be random. So there can't be any sort of preference or any sort of sexual selection for particular phenotypes over others. There can be no mutation of the alleles. We also can't have individuals coming into the population or leaving the population. And then finally, there can be no selection. So no natural selection, no artificial selection, every genotype has to have an equal chance of passing on those alleles to the subsequent generation. So any of these conditions, if they aren't met, is going to count as a disturbing force. So a disturbing force could be a population that's too small. A disturbing force could be non-random mating, mutation occurring, which mutations will occur if DNA is replicated. So this one is always going to be broken. Mutations occurring is a disturbing force. Differential migration is a disturbing force. And if there's any kind of selection, so natural selection occurring, artificial selection occurring, those would be disturbing forces. And so, other way. When we're thinking about Hardy-Weinberg equilibrium, if these disturbing forces are present, then Hardy-Weinberg equilibrium isn't going to be happening. So you might be asking yourself, what is the purpose of Hardy-Weinberg equilibrium if it's not going to be an actual situation that's happening in a real population? And the answer is that we can use Hardy-Weinberg estimates of genotype and allele frequency to get sort of a baseline to compare our population against. So we have two equations that can come into play if Hardy-Weinberg equilibrium is true for a population. And you can see these two equations here. They're P plus Q equals 1 and P squared plus 2PQ plus Q squared equals 1. And the idea is that if a population is in Hardy-Weinberg equilibrium, so if there's no evolution happening and there's no change in genotype or allele frequency, then we can use these equations to estimate allele frequencies and genotype frequencies. We can then compare these frequencies that we calculated to the actual population that we're looking at and see if they're similar or not. If the Hardy-Weinberg calculations and our actual population genotype frequencies and allele frequencies are very, very similar, then we can say, oh, it looks like these numbers that we calculated for this is what we would expect to see if no evolution was happening, is actually pretty similar to what our population looks like. So maybe evolution isn't really, there's not a lot of evolutionary force on this population. There's not a lot of selective pressure, or maybe the population's really big, but it doesn't seem like the population is evolving very much. But if we calculate these frequencies using Hardy-Weinberg equations, and then we compare them to our actual population and find that there's a very big difference. So our estimated genotype and allele frequencies are not at all similar to what we actually see in the population. Then we can assume that there is evolution happening in this population, depending on how many generations have passed, perhaps quite quickly. And then we can start to investigate maybe there are specific selective pressures, maybe there's natural selection, maybe there's genetic drift, but what's at play to be causing this evolution in the population? So Hardy-Weinberg gives us a sort of baseline to compare our populations against in order to see if evolution is happening or not. So the rest of this presentation will just be going into a little bit more detail about the equations that are involved with calculating these Hardy-Weinberg equilibrium genotypes and allele frequencies. So let's take a look at these two equations. The Hardy-Weinberg equations are going to use P's and Q's as opposed to capital letters and lowercase letters to represent dominant and recessive alleles. So normally we would say something like big A and little a, and big A is dominant over little a. But for Hardy-Weinberg, just to keep everything consistent, because sometimes it's going to be a big A and a little a, sometimes it's going to be a big C and a little c, sometimes big F and little f. Instead of dealing with different letters, capitalized and not, instead we just use a P and a Q. And the P is always going to represent the dominant allele frequency, so how common that dominant allele is, and this is this P right here. And the Q is going to represent the frequency of the recessive allele. So that's this right here. So if we had a population where there was a big A and little a, and the big A represented 80% of the population and the little a represented 20, then P would be 0.8 and Q would be 0.2. But always these frequencies in the decimal form should add up to 1. So we need the dominant and the recessive allele to together make up 100% of the population, or 100% of the alleles that we can have. So this first equation, P plus Q equals 1, is going to represent our allele frequencies, and the bottom equation is going to represent our genotype frequencies. So the first term in this equation is P squared, and really P squared just means P times P, right? And we said that P was our dominant allele. So P squared is going to represent the frequency of our homozygous dominant individuals. 2PQ has a dominant allele and a recessive allele. So PQ is going to be the frequency of heterozygotes, and Q squared is going to be the frequency of our homozygous recessive individuals. And again, all these different kinds of individuals with each genotype should add up to 100% of the population. So let's look at an example of how we can use these equations in a problem. So this problem is looking at a population of centaurs, and they can exist in either a brown color or a white color. And for this example, the brown color is dominant over the white color. And we have 75 brown-coated centaurs and 25 white-coated ones. So the thing to keep in mind, we're trying to find P and Q, and I've put our two equations over here. So the thing that we need to keep in mind with Hardy-Weinberg is that for any problem, we know for sure the genotype of the recessive, the ones with the recessive trait. So the ones that have white coats, we can be sure of their genotype, but the ones with the brown coats, we can't be sure what genotype they are. And that's because since the brown is dominant, they could either have two dominant alleles, capital B, or they could be heterozygous, so big B, little b. And either of these situations would result in a brown coat. And I've sort of put in parentheses, instead of using our big B, little b to signify these genotypes, I've put it in sort of like Hardy-Weinberg terminology. So two dominant alleles together is going to be our P squared, and big B little b is going to be PQ. In contrast, if an organism has a recessive phenotype, so in this case, like being a white centaur, we know what their genotype has to be because they have to have two recessive alleles to show that phenotype. So little b corresponds to Q squared, and we can be sure that all of the ones that are white are little b. So what we're going to do is we're going to calculate the frequency of this white coloration, which therefore we're calculating the frequency of how many centaurs have a little b genotype. So there are 25 white centaurs out of a total of 100. So the frequency of little b, or Q squared, is going to be 0.25. So we can see that here, Q squared is the white phenotype, or little b, still the same thing, and that frequency is 0.25. So if we take the square root to find Q, then we're saying Q is equal to 0.5. So if you take the square root of 0.25, you get 0.5. And then finally, so we found one part. We need Q and P. We found Q. And then the only thing left is P. And if we look at this equation right here, we know that P plus Q have to add up to 1. So we know that P has to be, I just covered it up, P has to be 0.5 as well, because if Q is 0.1, then we could do 1 minus 0.5 to get a P value, or to get a number for P that is 0.5 as well. Okay, we're going to do a lot of practice with these different kinds of Hardy-Weinberg problems in class. So practice makes better for these kinds of problems. So the easiest way to get better and more comfortable with doing these is to just practice problems, which we will do in class. So I will see you all later.